Hello, my name is Joshua Mugabe. I welcome you to Cell Respiration. Our today's objectives, we shall look at cell respiration. We shall look at adenosine triphosphate, anaerobic respiration, aerobic respiration, fermentation, sources of energy, and lastly, respirometry. You are welcome. Please do not forget to subscribe. Thank you. Cell respiration. Cell respiration. Cell respiration is the controlled release of energy from organic compounds. Cell respiration is the controlled release of energy from organic compounds, and principally it is glucose to produce ATP within the cell. That's what we call cell respiration. The release of energy from organic compounds. The organic compound, usually it is glucose, and we end up producing ATP within the cell. The organic molecules release energy or store energy within their chemical bonds. So during cell respiration, we are going to use the organic molecules, and those organic molecules have stored energy in their bonds. The energy is not easily accessible. This energy is not easily accessible for use in the cells. So the energy is stored in the bonds of the organic molecules, but it is not easily accessible for use in the cells. So the ATP functions as an intermediate source of energy within the cell. So it is the ATP which is going to function as the intermediate source of energy in the cell, and the energy now is readily accessible for use within the cell. That is now our ATP. ATP is the energy which is readily accessible to be used within the cell. So we have the glucose molecule. This glucose molecule is our organic substance, which we are going to break down, and then it will give us the energy which is accessible for use in the cell, which is ATP. And that is the energy that we are going to be able to use within the cell. But when the energy is still in the organic compound, in the bonds of the organic compound, it is not accessible. We cannot access it when it is in the organic compound. So we have to break it down, and then it gives us what we are calling ATP. Now the energy in ATP is the one which is accessible, and it is the one that we are going to use in the cell. Now the ATP is considered to be as the energy currency of the cell. That ATP that is produced from the breaking down of the glucose, that ATP becomes what we call the energy currency of the cell, meaning that it is the one which is accessible to be used by the cell. The cell respiration is controlled release of energy from organic compounds to produce ATP. So when we break down organic compounds and we produce ATP, that process is what we call cell respiration. It is a controlled release of energy, a controlled release of energy from organic compounds to produce ATP. And our organic compounds always we use is glucose. Glucose is the organic compound. So the release of the energy which is stored in the bonds of glucose to give us ATP, That process is what we refer to as cell respiration. Now let us look at adenosine triphosphate. Let's look at adenosine triphosphate. Now ATP is a high energy molecule. ATP is a high energy molecule, and it serves as an intermediate energy source in the cell. That is ATP. ATP is an abbreviation of adenosine triphosphate. It is what we refer to as ATP, and it is a high energy molecule that serves as an intermediate energy source within the cell. It consists of nucleosides. It is consisting of a nucleoside linked with 3 phosphates. It is linked with 3 phosphates via high energy bonds. Now I want us to look at our nucleoside. The nucleoside we are talking about is this one, which is made up of a ribose sugar and adenine base. That is what we are referring to as a nucleoside. And this nucleoside, we are saying that it is linked to 3 phosphates. The 3 phosphates that we are talking about, they are these 3 phosphates here. Those are the 3 phosphates the nucleoside is linked to. And they are linked via high energy bonds. So you can see that from one phosphate to another phosphate, there is a bond. That bond is a high energy bond, which is linking those 3 phosphates. So when ATP is hydrolyzed to ADP and an inorganic phosphate, the energy contained is released for use. So we have our ATP molecule, which we are referring to as here, the ATP molecule. When it is hydrolyzed is being broken down, and it will turn into what we call ADP. Now ADP is what we refer to as adenosine diphosphate. Now the adenosine diphosphate is this molecule that we have here, our ATP, which has been hydrolyzed. By hydrolyzing it, we are removing one inorganic phosphate. One inorganic phosphate is broken off. And when it is broken off, we say that what remains behind is what we are calling ADP, adenosine diphosphate, meaning that it has only 2 phosphates remaining. One has been broken off. And when that one is broken off, then we release the energy that is contained in that bond. That bond which broke off the one phosphate, that bond released the energy, and it is this energy which is released is the one that is going to be used in our cell. So when we hydrolyze ATP to ADP, we get energy which is available for use in the cell. Let's look at the types of cell respiration. Cell respiration can involve 2 distinctive mechanisms. It can involve 2 distinctive mechanisms. There is one we call anaerobic. Now in anaerobic mechanism, there is partial breakdown of organic compound for a small ATP yield. So what we are saying that is that in anaerobic respiration or mechanism, there is partial breakdown. The organic compound is partially broken down. And when it is partially broken down, it results in a small ATP yield. We get little amount of ATP. For example, here we have our glucose molecule. When this glucose molecule is broken down in the absence of oxygen, anaerobic means absence of oxygen. If there is absence of oxygen, we shall get pyruvate, 2 pyruvates. Now these 2 pyruvates are now the ones which are going to be to use the 2 hydrogen carriers. When they use the 2 hydrogen carriers, then we shall get 2 ATP. Little amount of energy is going to be produced. That is during anaerobic mechanism or anaerobic respiration. Then there is a second one which we call aerobic respiration. Now in aerobic respiration, it takes place in the presence of oxygen. Now here, the complete breakdown, it is the complete breakdown of the organic compound, and this one results in a large ATP yield. We end up producing a lot of energy. So here our glucose is going to be broken down in the presence of oxygen, and when it is broken down in the presence of oxygen, we shall get carbon dioxide, and then we shall get water, and we shall get a lot of ATP. 36 molecules of ATP are produced. So those are the two mechanisms in which cell respiration can undergo. We have aerobic and anaerobic respiration, and we are seeing that aerobic respiration takes place in the presence of oxygen, and we produce high molecules of ATP, 36 molecules. And the anaerobic, we saw we produced only 2 ATP molecules. Let's now look at anaerobic respiration. Now, in anaerobic respiration, this involves the partial breakdown of glucose for a small ATP yield. We have already seen that one. That in anaerobic respiration, there is a partial breakdown of glucose. It is not completely broken down, but it is partially broken down, and we end up yielding a small ATP molecule. So the net gain is only 2 ATP molecules that we are going to get from anaerobic respiration. And this one occurs via a process called glycolysis. And glycolysis takes place in the cytosol. Cytosol is the same as the cytoplasm. Now let us look via glycolysis. What glucose is converted into pyruvate. We are having our glucose is being converted into pyruvate. Remember, we say that it takes place in the cytoplasm. So here we have our glucose molecule. If we look here, we have our glucose molecule here, 6 carbon atom, and this 6 carbon atom is going to be converted into pyruvate. Pyruvate, there are 3 carbon compounds. Those are the ones that we call pyruvate. So our glucose is now converted into the pyruvate, and there is a net gain of 2 ATP. Here you can see that we have got 2 ATP. We produce 2 ATP, that is during glycolysis. 2 ATP are produced, and then oxidized carrier molecule NAD plus are reduced to 2 hydrogen carrier molecules. So you can still also see now here we have 2 oxidized carrier molecules. They will end up producing 2 hydrogen carrier molecules. And if you look very carefully here, you will see that we are also having ATP here. When we have ATP, then we shall also use 2 ATP molecule. We have used the 2 ATP molecule to get 2 ADP molecules. Now, the total production of energy that we are going to produce, we shall produce 4. We produce 4 ATP molecules. At the end of it, we end up producing 4 ATP molecules. But remember, in the beginning, we used the 2. So if you have the 2, these ones we used, and you subtract them from the 4 we used, then a net gain is going to be 2 ATP that we are going to have to pay off. That is going to be our energy payoff. There will be only 2 ATP. So we are saying that glucose is converted into pyruvate, 2 of them, and the net gain is only 2 ATP. We have seen that one. Here we say that we had to use the 2 ATPs. In the beginning we used 2 ATPs, and at the end how much did we produce? We produced 4 ATP. So if you have 2, you use the 2, and at the end you produced 4, that means that you use the 2, so you have the net production will be only 2 ATP. And there we have seen, we have used the oxidized carrier molecules, NADP plus, and those ones are reduced to form 2 hydrogen carrier molecules, which are NDPH. Let's now look at aerobic respiration. In aerobic respiration, this one involves the complete breakdown of glucose. It is a complete breakdown of glucose, and it leads to a higher or large ATP yield. Here the net gain of ATP is 36 molecules of ATP are produced, and that one happens during aerobic respiration. Remember aerobic respiration, that's where we have said we use oxygen. There is a presence of oxygen. And when there is a presence of oxygen, it means that we undergo complete breakdown of glucose and we produce a lot of ATP, and we end up yielding 36 molecules of ATP. Now this one requires oxygen. Anaerobic respiration requires oxygen, and it takes place in the mitochondria. Now let us look at aerobic respiration. What happens during aerobic respiration via aerobic respiration? Aerobic respiration involves complete breakdown of glucose, and we end up producing large quantities of ATP, which ends up producing 36 molecules of ATP. And we have said that this one takes place in the mitochondria. And then we also end up producing hydrogen carrier molecules are made in large quantity. So we look here in anaerobic respiration. Now anaerobic respiration, we said it takes place in the cytoplasm. Takes place in the cytoplasm. That's where it stops. And that process is what we are referring to as a glycolysis. Glycolysis is that process which results in anaerobic. In anaerobic respiration, or what takes place in the cytoplasm, that is glycolysis. Glucose is broken down into pyruvate. That one does not require oxygen. And in that one, we end up producing only 2 ATP molecules. We produce only 2 ATP molecules. Now, when the pyruvate moves from the cytoplasm, it enters into the mitochondria. Now, in the mitochondria, there we shall need energy. We shall need, oxygen. They need to use oxygen. And now this one will go into the link reaction. It will go through the link reaction. Then from the link reaction, we shall go to the citric acid cycle. And in all those processes, we are producing a lot of hydrogen carriers. A lot of hydrogen carriers are produced. And when we produce a lot of hydrogen carriers, the more the ATP molecules we are going to produce. We shall produce a lot of ATP molecules. And that one is what we are referring to as aerobic respiration. Aerobic has both the cytoplasm, it occurs both in the cytoplasm and also in the mitochondria. Now, aerobic respiration, these hydrogen carrier molecules, the NADH, are used to produce significant amount of ATP. That is a net of 36. And this one is via a process we call oxidative phosphorylation. So we are having our hydrogen carrier. All these ones that we have here, they are the hydrogen carrier molecules. And we are saying that these hydrogen carrier molecules are used to produce a significant amount of ATP. And so you can see that here, every hydrogen carrier molecule is producing an amount of ATP, which is our energy. And we end up with a net of 36 ATP. If you count, these are 22. 22 plus 2, those are for 24. 24 plus 6, that one becomes 30. 30 plus 4, 34. And then 34, this 4, if you add on the other 2, it becomes 36. So we end up producing a net of 36 ATP. And this one is via, that whole process is called oxidative phosphorylation. It's what we refer to as oxidative phosphorylation. Now let us look at anaerobic respiration versus aerobic respiration. Now we look at the reactants. What are the reactants in aerobic respiration? In aerobic respiration, you'll see we are using glucose. Anaerobic respiration, we use glucose as the reactant. And in aerobic respiration, we use glucose. At the same time, we use oxygen as the reactant. Now, when it comes to combustion, this one is partial. In anaerobic respiration, the combustion is partial, meaning that it is incomplete, whereas in aerobic respiration, the combustion is complete. When it comes to energy yield, we yield in anaerobic low energy yield. Only 2 ATP are produced, that is during anaerobic respiration. 2 ATP, which is low. And in aerobic respiration, we produce 36 minus 38 ATP. 36 minus 38 ATP. Though, so the net production is 36, though what is produced is 38. Remember, when we talked about glycolysis, we first use 2 ATP. And those 2 ATPs, when we produce them, we end up producing 4 ATP. So we have to subtract those 2 to pay back. Now, when it comes to the products in anaerobic respiration, we produce pyruvate, which is then fermented. That is during anaerobic respiration. Whereas in aerobic respiration, we produce carbon dioxide and then water. Those are the products. Now, when it comes to the location, anaerobic respiration takes place in the cytosol. Cytosol is the same as the cytoplasm. And the aerobic respiration occurs both in the cytoplasm or the cytosol and the mitochondria. Let's look at the fermentation process. Now, during the fermentation process, glycolysis involves the formation of reduced hydrogen carrier molecules. We produce NADH. That is in fermentation. Remember, in the fermentation process, this is anaerobic respiration. It is taking place in the cytoplasm, or what we refer to as via glycolysis. So during this process, there is formation of hydrogen carriers. Now, these carriers are used in aerobic respiration. They will be used in aerobic respiration to produce a large amount of ATP. Now, when NADH are produced during glycolysis, they will be carried to the aerobic respiration. In aerobic respiration, that means that we need the use of oxygen. So they will be transported from the cytoplasm to the mitochondria, where we shall use now oxygen, and then we shall produce a lot of ATP. Now, in the absence of oxygen, respiration occurs anaerobically. Only glycolysis occurs. So in absence of oxygen, what is going to happen? Respiration is going to stop only in the cytoplasm. That is what we are referring to as glycolysis. Glycolysis process is part of the respiration. But for it, glycolysis takes place in the absence of oxygen. It does not require oxygen. So if we don't have any more oxygen, then that means that the process will not continue going or will not take place in the mitochondria. So it will only stop in the cytoplasm, and it will only stop at the glycolysis process. That is, if there is the absence of oxygen. Respiration will occur only anaerobically. That is glycolysis occurring only. So this exhausts the supply of oxidized precursor of the carriers, that is NADPH. So if fermentation is taking place, now fermentation is taking place, it is only stopping in the cytoplasm. So it is only glycolysis which is taking place. And if glycolysis is taking place, means that we are breaking down glucose and breaking it into pyruvate. So if you break it into pyruvate and it stops there, does not go into the mitochondria, then it means that we are going to become exhausted of the oxidized precursor. If that one happens, then glycolysis will stop. So what happens? Fermentation involves the reversible conversion of pyruvate into an intermediate form via the oxidation of hydrogen carrier molecules under anaerobic condition. So here what we are looking at fermentation, and we are saying that this one involves a reversible conversion of the pyruvate into an intermediate form via the oxidation of the hydrogen carrier molecules, and this one is undergoing under anaerobic condition. Now here, remember we are saying that we are in the cytoplasm or cytosol. We are breaking down glucose to pyruvate. If you break down glucose to pyruvate, that means that it will stop there. So how can we make sure that it continues? Remember, when we broke down glucose to pyruvate, we produced 2 hydrogen carriers. And if those hydrogen carriers are there and they are not used, then it means we have exhausted our oxidized carrier molecules. So for the process to continue, we need to get oxidized carrier molecules. But the oxidized carrier molecules have been converted into the hydrogen carrier molecules. So what should we do to avail the oxidized carrier molecules? So this is what is happening. Fermentation is going to take place. Now what we are going to do, we are going to turn our pyruvate. The pyruvate now is going to be converted into an intermediate form. And when it is being converted into this intermediate form, it will have to use the hydrogen carrier to be converted into an intermediate form. And when the hydrogen carriers molecules have been utilized, then that means now we shall end up releasing the oxidized carrier molecules. And when the oxidized carrier molecules are released, then that means now we can now break more glucose to pyruvate. So this restores the stock of the precursor NAD, that is now our fermentation, and allowing glycolysis to continue. So we are saying we are restoring the stock of the precursor oxidized carrier molecules. Remember, we had produced the hydrogen carrier molecules and we had exhausted the oxidized carrier molecules. So when we use the hydrogen carrier molecules to convert the pyruvate into an intermediate molecule, then the hydrogen carrier molecule are converted back to the oxidized molecule, carrier molecules. And when they are turned back to the NAD+, then now glycolysis can continue. Now, fermentation, we say that for it, it involves a reversible conversion of pyruvate, whereby pyruvate is converted into an intermediate form. And this one is via the oxidation of the hydrogen carrier molecules. So when we convert these carrier molecules, the hydrogen carrier molecules, if we have the hydrogen carrier molecules and we are oxidizing them, they will go back and form what we are calling our oxidized carrier molecules. And when we get these oxidized carrier molecules, then now it means that now glycolysis will continue. So fermentation in animals. Let's look at fermentation in animals, if it is taking place in animals. Now, fermentation in animals includes pyruvate being converted into lactic acid. So I want us to look at it here very closely. We are saying in fermentation, it is taking place in the cytoplasm. And the process is glycolysis. The process is glycolysis. So we are having our glucose. The glucose molecule here is converted into pyruvate. When we convert our glucose molecule into pyruvate, we are going to produce ATP, and then we shall convert the oxidized carrier molecule is going to be converted into hydrogen carrier molecule. So now here, when this one happens, it means that if we still have pyruvate, we cannot now break down any more glucose. So what we have to do, we have to remove this pyruvate. And this pyruvate is now going to be converted into lactate. But for it to be converted into lactate, it means that we have now to use the NADH, which is our hydrogen carrier molecule. The hydrogen carrier molecule will have to be oxidized to the oxidized carrier molecule, which is NAD+. Now, when we form NAD+, after converting pyruvate into lactate, now we have removed the pyruvate and we have also produced the oxidized carrier molecule. Then now we can break down more glucose to pyruvate. So that is what happens in humans or animals. The glucose is converted into pyruvate, and the pyruvate is now converted into lactate, which lactate is in the muscle, and now we can continue with the glycolysis process. The glycolysis process can continue. That is what we are calling fermentation in animals, or glycolysis taking place in animals, or a reaction which is taking place in the absence of oxygen. And this one will help us to produce some more energy, but the energy will be little. Little energy is going to be produced in this process. So we are saying that what is going to happen, the build up of lactic acid within the muscle is going to lead to muscle fatigue. So when we are converting our glucose here, when you convert this glucose and then you convert it into pyruvate, this pyruvate is going to be converted into lactate. And when it is converted into lactate into the muscle, now that means that it is going to cause what we are calling the muscle fatigue. It is going to cause the muscle fatigue. And what is causing the muscle fatigue is because there is high accumulation of lactic acid, or what we are calling lactate, in the muscle tissue. That one is going to lead to what we are referring to as a muscle fatigue, because when we are breaking down glucose, it is incompletely broken down. we could break it down to pyruvate, but because there is no oxygen, it will be converted to lactate, which is stored in the muscle of this athlete of ours. Now, when it accumulates in the muscles of this athlete, now that one is the one which is going to cause what we are referring to as muscle fatigue. So muscle contraction requires energy. The muscle contraction requires energy. So when our muscles are doing work, they are contracting. And that contraction of the muscle to do work, it means that they require a high level of ATP, energy. And that one will mean that we need aerobic respiration to take place. So during the strenuous exercise, that is when you are doing a lot of exercise, there is going to be an oxygen demand by the muscle. So when you are doing a lot of exercise, like for example, this gentleman of ours here, who is doing a lot of exercise, his muscles are going to require a lot of energy. And when they require a lot of energy, the muscles, now they will outstrip the supply, meaning that the amount of energy which is needed by the muscle is now demanding a lot of oxygen. And if it is demanding a lot of oxygen, this one now will exceed the amount of oxygen being produced. And if that one happens, now there is less oxygen, then that means that now our pyruvate is going now to be broken down in the absence of oxygen. And if it is broken down in the absence of oxygen, it will now be converted into lactate. And when it is converted into lactate, now it is going to be stored in the muscle. And if it is stored in the muscle, or accumulation of a lot of lactate in the muscle is going now to cause what we are calling the strain in the muscle or pain in the muscle. So the lactate production in humans uses anaerobic respiration to maximize the power of muscle contraction. So when we produce lactate in humans, this process is taking place anaerobically. When we are producing lactate, it means that the process is anaerobic. There is no oxygen being provided. And this is done so to maximize the power of the muscle contraction. For example, we are saying, this gentleman of ours here is doing a lot of running, but there is less supply of oxygen in the muscle. So what is going to happen? Glucose is going to be broken down to lactate, and the lactate needs to be broken down. Further breakdown of lactate will require oxygen, but there is no oxygen being supplied. And if oxygen is not being supplied, but we still need to produce energy in the form of ATP, but there is no oxygen. And if there is no oxygen, that means that pyruvate has to be broken down in another way. So it will be now broken down into lactate. And if it is broken down into lactate, then we shall continue with that cycle, glucose being broken down to pyruvate, pyruvate being broken down to lactate, so that we produce some little amount of energy, which is going to provide the power of muscle contraction in our gentleman here due to lack of oxygen. But when that one is happening, there is a lot of lactate which is being stored in the muscle. And if it is stored in the muscle, now that one is what we are referring to as anaerobic respiration, or what we refer to as fermentation. Thank you very much for participating. Please don't forget to subscribe. So fermentation in plants. Fermentation in plants and yeast. In plants and yeast, pyruvate is fermented to form ethanol and carbon dioxide. So we have looked at fermentation in animals where we produce lactate. But when it comes to plants and yeast, our pyruvate is going to be fermented into ethanol and carbon dioxide as the end product. And this one can be used, for example, in the preparation of soya sauce. There is lactic acid. And then there is in the preparation of yogurt and cheese. That is still anaerobic respiration. Still there is lactic acid. Now when it comes to our beer, there is production of ethanol and carbon dioxide. Wines, there is ethanol. And then in breads, that is we have carbon dioxide. So fermentation in plants and yeast, it ends up yielding ethanol and carbon dioxide. Sources of energy. Now, cell respiration typically relies on carbohydrates. All respiration in the cell, that is cell respiration, typically relies on carbohydrates as the initial source of energy. But other organic molecules, there are other organic molecules that may also be digested to produce ATP. Carbohydrates can be digested anaerobically. Carbohydrates, for them, they can be digested anaerobically. Whereas triglyceride, these ones produce roughly twice as much energy, but this requires aerobic digestion. Now let us look at our diagram here. When we look at our diagram here, we can see that we have glycolysis here. Glycolysis is taking place. But when glycolysis is taking place, the energy source is going to be carbohydrates. It is mainly carbohydrates that occur in glycolysis. And glycolysis does not need oxygen. So when you look at the triglyceride cannot all of them go into glycolysis. But only triglycerides, they are going to be broken down. And when they are broken down, they will be broken down into glycellose, and then they will be broken down into fatty acid. And when they are broken down into glycellose and fatty acid, those are the ones, now glycellose are the ones that can be converted to glycolysis. The fatty acids will be taken into the link reaction. Now, that is what we are saying, that carbohydrates can be digested anaerobically, meaning that they are the ones that only undergoes glycolysis if it is to stop there. But triglycerides cannot go through glycolysis because the fatty acids cannot pass through that route. Now the proteins, for the proteins, these ones, digestion produces nitrogenous wastes. If you look at the proteins, our proteins will end up producing what we refer to as nitrogenous wastes. So when you look at the proteins here, the proteins, they produce what we call the amino acids. Proteins are broken down into amino acids, and the amino acids, they produce nitrogenous wastes. But the amino acids can be taken into the link reaction, and then they can also be taken directly into the citric acid cycle. So where you are seeing this color means that there the process which takes place there is anaerobic, does not need oxygen. And where you are seeing this color, it means that there we need oxygen, therefore it is aerobic. So from glycolysis here does not require oxygen. So the reactions which are taking place here are anaerobic. But when you cross from glycolysis and you enter into the link reaction, the ring reaction here requires energy. Now the reactions that are taking place beyond here, these ones are anaerobic respiration, or they require energy. Let's look at our respirometry. Now, a respirometer is a device that determines the organism respiration rate by either measuring oxygen uptake or carbon dioxide production. That is our respirometer. It is our device that we are going to use to determine the organism's respiratory rate or respiration rate. And this one can either be determined by measuring the amount of oxygen it takes in or the amount of carbon dioxide it is producing. So what we have here is our respirometry. The whole of that one is our respirometry. All of this one is our respirometry. So it is the one that we are going to use. And here we are having our specimen, which we want to measure the amount of oxygen it is taking in and the carbon dioxide it is producing. Here we have the specimen and the carbon dioxide absorber. And here we have a source of air. Also there is equal mass of volume, okay, of air. So now it will pass through, and here in the middle we have our tube which is having water. So the specimen respires, can cause the pressure change, moving the water between the platform. Of course, when this one is respiring, it will produce the gas, which gas will come in here and mix the water. And that one may force the water to move from this side going the other direction. So when water moves in the other direction, this one means this side it is going to go down and this side it will go up. So that one can be noted. Change in the pressure. Now carbon dioxide being produced is going there and changing the pressure. And then also carbon dioxide can cause the change in the color of the water because it is dissolving there and forming an acid. Or when this one is taking in oxygen, we can also expect this water to rise as oxygen is being removed. So we expect the water to rise going up this side, and this side it will be reducing going downwards, meaning that oxygen is being removed, and as oxygen is being removed, that means that they're going to cause the water to rise up this side. It will rise, whereas this side the water will be going down. So oxygen uptake can be measured by pressure. We can use pressure change in the manometer. So when you take in oxygen, this specimen, when this specimen takes in oxygen, what is going to happen? What will happen will be that when it takes in oxygen, that means the oxygen in the tube is going to go into the cylinder. And as the oxygen is going into the cylinder, this oxygen which is going to the cylinder, it is going to pull the water, and the water level now this side is going to rise. And the water level this side is going to go down. Therefore, oxygen uptake can be measured by the pressure change in the manometer. Carbon dioxide production can be measured by change in the pH. It can be measured by change in the pH in the water. That is, carbon dioxide will form what we are calling carbonic acid. So if now it is producing, our specimen here is producing carbon dioxide, what is going to happen is that this carbon dioxide will pass through the tube and then it will enter into the water here. So when it enters the water here, it is going to react with water to form carbonic acid, which is going to lower the pH of the water, and when it lowers the pH of the water, then we are able to see the change in the color of the water. So that's how we can measure the production of carbon dioxide. Thank you very much for participating in cell respiration. And during this process, we have talked about cell respiration. We have discussed cell respiration. We have looked at adenosine triphosphate and anaerobic respiration. We have looked at aerobic respiration. We have looked at fermentation. We have looked at the sources of energy, and we have looked at the spirometry. Thank you. Please do not forget to subscribe.